Your handwriting is not a style choice. It is a biological readout. Every pressure variation, every letter formed too small or too large, every shaky baseline and fluid cursive loop is the surface expression of something happening deep inside your neural architecture. Scientists have spent decades trying to decode this signal. What they found was not what anyone expected. Very small, uniform handwriting. The first pattern is handwriting that is consistently, uniformly small, letters reduced far below a person's natural scale, controlled and precise in their miniaturization. More than 65% of people with Parkinson's disease develop this pattern, known clinically as micrographia. The term was coined in 1903 by Arnold Pick after observing it in a patient with a thalamic lesion. The underlying cause is damage to the basal ganglia, the subcortical structures responsible for regulating movement, amplitude, and automaticity. When the basal ganglia fail, the brain loses its ability to calibrate the force and scale of motor output. The letters shrink because the neural signal driving the hand weakens. Dopaminergic medication can restore this signal in some patients by rebuilding the cortical basal ganglia thalamocortical circuit. In individuals without neurological disease, consistently small handwriting reflects the same circuitry operating at lower output, a motor system that is functioning, but functioning conservatively. The brain is precise. The movements are controlled. Small handwriting is not a sign of shyness or introversion. It is a signature of tightly regulated motor circuitry in the basal ganglia. Handwriting that shrinks as it continues. The second pattern is more specific and neurologically more revealing. A person begins writing at a normal size, but as the line continues, each word becomes progressively smaller, as if the brain is running out of signal mid-sentence. This is progressive micrographia, and it maps onto a fundamentally different neural failure. Researchers using fMRI found that this pattern correlates with disconnections between the rostral supplementary motor area, the rostral cingulate motor area, and the cerebellum. These structures govern the sequencing and sustained execution of complex motor programs. When their connectivity degrades, a motor action can begin normally, but cannot maintain its calibration over time. Critically, the dopaminergic drugs that help consistent micrographia have no significant effect here, because the breakdown is not in the basal ganglia circuit, but in the supplementary motor area's relationship with the cerebellum. Two patterns of shrinking handwriting. Two completely different neural failures. The pen is rendering a connectivity map in real time, drawn across the page, shaky and trembling strokes. The third pattern, trembling, unsteady strokes that lose their path, is one of the earliest documented signals of Alzheimer's disease. This alteration appears before many other clinical symptoms become apparent. What looks like poor motor control is the early expression of neurodegeneration affecting muscle regulation, spatial processing, and memory simultaneously. As the condition progresses, the deterioration deepens from shakiness into illegibility, eventually reaching language systems and producing phonological spelling errors. In 2025, researchers analyzed handwriting samples from 174 participants, 89 Alzheimer's patients, and 85 healthy controls, and used artificial intelligence to distinguish between the two groups. Their accuracy reached 93.33%. Their sensitivity reached 99.110%. The dataset, called Darwin, is the largest publicly available handwriting collection for Alzheimer's research. The hand does not lie about what the brain is experiencing. It simply reports it in a form most people mistake for age or fatigue. Heavy pressure and deep, emphatic strokes. The fourth pattern is heavy pen pressure, handwriting pressed firmly into the page, dark and emphatic, the indentation visible on the sheet below. This reflects high activation of the sensorimotor cortex and deep engagement of the haptic feedback loop. Sophia Vinci Bucher of Vanderbilt University has noted that the fingers must each do something different to produce a recognizable letter, and when someone presses harder, they amplify the tactile and proprioceptive signal sent back to the brain. The sensorimotor cortex responds to that pressure with increased engagement. The stronger the haptic feedback, the more robustly the brain's writing network is activated. Heavy writers are, neurologically, giving their brains more information to process with every stroke. The evolutionary logic is straightforward. The brain prioritizes tasks that generate strong sensory feedback, treating them as high stakes and encoding them more deeply. The trade-off is fine motor fatigue and reduced fluency over sustained writing periods. But the neural engagement is measurably higher. Cursive and connected writing. The fifth pattern is cursive, fluid, connected letter forms where the pen rarely lifts from the page. This style engages the motor cortex and cerebellum more intensively than disconnected printing, because cursive demands continuous coordination of smooth transitions between letters. The cerebellum manages real-time fine motor correction across an unbroken sequence of movement, making sustained cursive one of the more cerebellar demanding everyday tasks a person performs. Research has found that cursive can preferentially increase writing and reading speed and fluency, particularly for individuals with dyslexia or dysgraphia. The connected nature of the script reduces the motor interruptions that block letters create, allowing language to flow with less cognitive overhead. Dr. Corinne James of Indiana University, whose MRI research established the neural reading circuit built by handwriting practice, found that the haptic feedback generated by forming letters by hand is the central driver of this benefit. Cursive amplifies that feedback through continuous movement. However, when James scanned brains comparing cursive directly to print, she found no fundamental difference in neural activation between the two styles. The form, it turns out, matters less than the act of forming it. Deliberate separated block printing. The sixth pattern, intentional separated block printing, carries its own distinct neural signature. James's landmark research with pre-literate 5-year-olds demonstrated that children who practiced printing letters by hand activated a reading circuit in the fusiform gyrus, inferior frontal gyrus, and posterior parietal cortex that tracing and typing completely failed to produce. She described the left fusiform gyrus as the convergence point, where visual stimuli become letters and written words. Printing, done deliberately and formed by hand, constructs this circuit in the developing brain. The slight imperfections in each hand-drawn letter, the natural variability of human motor output, are precisely what triggers the neural recognition response. Perfectly uniform digital type bypasses this learning mechanism entirely. The brain builds its reading architecture by building its writing architecture first. Irregular and inconsistent handwriting. The seventh pattern is handwriting that lacks consistency across a single page. Letters form differently each time. Pressure varying mid-sentence. Baseline wandering without anchor. The 2024 NTNU study found that optimal brain connectivity during handwriting manifests as widespread, synchronized activity across parietal and central regions. When motor output lacks consistency, it reflects weaker integration across the sensory motor network, a less precisely encoded internal model of each letter. The hand's output is only as stable as the neural program driving it. Irregularity is not creative expression. It is a lower resolution motor signal, reflecting a writing network that has not reached full encoding depth. Rapid, automatic. The eighth pattern is speed. Handwriting that flows quickly. Letters formed without conscious attention. The pen moving as if thought itself has taken physical form. This reflects motor automaticity. The basal ganglia have internalized the motor program so completely that conscious oversight is no longer required. The left putamen, a structure within the basal ganglia, governs this kind of learned automatic motor sequence. A 2018 study of 42 Parkinson's patients found that 6 weeks of intensive handwriting training produced measurable changes in neural connectivity within the writing network, particularly in the supplementary motor area. That finding inverts the typical narrative. It means handwriting is not merely a reflection of brain structure. It is a tool for rebuilding it. The written record of Uhu Yar. For over 4 centuries, human beings have believed that handwriting reveals character. Jeffrey Dean's meta-analysis of approximately 200 studies on graphology concluded otherwise. Graphologists failed to demonstrate validity for predicting personality or performance, and the British Psychological Society ranked the practice alongside astrology. Zero validity. And yet over 80% of European companies have historically used it in hiring. The instinct was not entirely wrong. The interpretation was, handwriting does reveal something real and measurable, not personality, not trustworthiness or ambition, or creativity, but the state of the basal ganglia, the connectivity of the supplementary motor area, the integrity of the cerebellum, the encoding depth of the fusiform gyrus. Every page you have ever written is a neural record you left behind without knowing it. The pen was always telling the truth. Science is now learning to read it.